So we talked about the beginning part of cells. Now we're going to talk about, we talked about cell theory. We talked about all those other things in the first part of this. Now we're going to talk about the eukaryotic cells. And what you notice here in the eukaryotic cells, as opposed to the prokaryotic cells, is that it is much more defined in what's going on inside of the cell itself. Okay? There are all the organelles that you can see, the nucleolus, the nuclear envelope here. No, it's not what I wanted. You see the nucleus, the nuclear pore, all of those things are showing up here. Mitochondria, microtubules, ribosomes, like all of these are going to be the main parts and the main organelles that are taking care of the cell. Every single thing has a very specific peroxisome, lysosome, the Golgi complex. They all have very specific roles in what they do. The centrosomes, the flagellum helps move the cell if it needs it. But again, you have your cytoplasm and you have your plasma membrane. All right. So when we talk about the plasma membrane itself, so here it is. It's a double lipid or phospholipid bilayer. Okay, so you have these heads that face outwards, which are hydrophilic, okay, in nature, which means if they're hydrophilic, what does that mean? What does hydrophilic mean? Yep. So that's going to attract the water, attract the H2O, right? So that's what it does. So the water molecules can come in and water can then pass through. It goes through the head and into the cell itself. These, the tails, are hydrophobic. Okay? They are water-fearing. God bless. So they're water-fearing. That means when the water molecule comes through, it's going to get pushed through all the way through so it goes into the cell itself. So you have this plasma membrane that it's made up of. Two layers. You can see the dividing right here, right? You can see the first set and the second set. So it's also made up of these glycoproteins, which you can see right here. It's sticking out of the membrane, the protein membrane as it is. So anytime you see these attachments, those blue attachments are glycoproteins. Okay? Glyco, what do you think? Yes, sugar proteins. So these glycoproteins are sugar proteins, okay? So they are a protein with a carbohydrate attached to it. The green ones are glycolipids, which means it's a lipid with a carbohydrate attached to it, okay? And these all help the cells gain and gather what they need to bring into the cell itself, okay? So you also have these peripheral membranes here. Okay, these peripheral membranes are on the outside, peripheral. What are we talking about? Peripheral vision. When you're, especially in athletics, we're talking about corners, right? We're talking about what you see out of the sides of your vision. So these are going to be on the outside of it. Okay, now these proteins have protein channels to bring in different types of molecules into the cell itself or to expel something out of the cell itself. And then also giving the plasma membrane some, I'm trying to think what the word is. Some structure would be the cholesterol, okay? And then these are the filaments of the cytoskeleton that are outside the cell as well. All right, so you'll see these embedded proteins. These embedded proteins are going to be the important piece to be able to bring things in and out of the cell itself, okay? So you can see, just so you see, so inside the small intestine, you have this microvilli that's on the plasma membrane. So what do we think has happened inside the small intestine? What goes on inside the small intestine? Digestion, exactly. So what this microvilli does is that it's, again, pulling material into the cells themselves to then, and then once it's been used, if there's any trash or anything that needs to come out of the cell itself, it is expelled out through the plasma membrane itself, okay? They increase the surface area. So anytime you hear microvilli or cilia or anything like that, anything that's like little appendages sticking off of the cell or off of an organelle, what it does is it allows for surface area to grow. They are only going, so these microvilli are only in the cavity that faces where the substances are coming from, okay? So you can see how these things are changed and how they're made. The cytoplasm, that's the cytosol that's inside that suspends the organelles and keeps them kind of in place for the rest of the cell. So the cytoplasm is the region between the plasma membrane and the nuclear envelope. The nuclear envelope is what surrounds the nucleus. This consists of the organelles suspended in a gel-like or gelatin-like cytosol, plus the cytoskeleton itself. 70 to 80% of the cytoplasm is water, but it has to have this semi-solid, solid's not the right word, it's almost like a gelatin, which I don't think any of us would ever, like, especially when gelatin first starts to set and it still is like really wiggly and stuff like that. So that's what the cytoplasm, that's the consistency of the cytoplasm. And it's because of the proteins that are in the cytoplasm that gives it that almost jiggly nature to it. I don't have another word for it. I can't think of another word for it. The nucleus. There's usually only one nuclei per cell, okay? And usually it's going to be the biggest organelle that's in the cell itself. I will say to you that, so remember the prokaryotic cells, and we looked at the capsules, they only had a nucleoid region. This nucleus could actually, for the most part, be larger than almost all eukaryotic or prokaryotic cells, okay? So you can see, here you see the nucleus and the pieces of the nucleus. So here you have that dark center spot for the inside the nucleus itself. That dark center spot right here is the nucleolus, okay? That's where a lot of things, it's kind of like the headquarters in the nucleus itself. You have the chromatin. What do you think the chromatin is involved in? What is it? What do we think chromatin? What do we think? So what's in the nucleus? DNA. Yes, DNA. So really, this is where we're thinking, okay, these are your DNA strands that are here that are ultimately going to be going into messenger RNA and things of that nature. So just like you have the cytoplasm in the outer cell, you also have this nucleoplasm that's inside the nucleus itself. Again, keeping things where they need to be, suspending them in that into the in the environment around and inside the nucleus. Here you have your nuclear pores where things can go in and out of the nucleus. And then you have the nuclear envelope, which goes all the way around, but is studded with pores to allow things to go in and out. And what do you see? Usually the nucleus will always be attached, in this case, to the rough endoplasmic reticulum, which you can tell because when you see the smooth endoplasmic reticulum, you won't see that it's studded with these ribosomes. All right, so it'll look much smoother than what that does right now. So the nuclear envelope, it's a double membrane, okay? It separates the DNA from the cytoplasm itself, and it separates transcription from translation. That's the big key of what the nuclear envelope does. That's its biggest function, is that it's separating transcription from translation, okay? And then the nuclear pores are the alleyways, for lack of a better term. They're the alleyways which will connect the nucleoplasm to the cytoplasm. It regulates the flow of molecules back and forth, just like the cell membrane does for the entire cell. These nuclear pores are controlling the flow of molecules back and forth out of the nucleus and into the cytoplasm, okay? NLS signals are needed, just like when you talked about, oh God, I just had it in my head and it went away. Diffusion of molecules across the cell membrane. You either have facilitated facilitated diffusion, which means energy is needed to be able to make the proteins bring materials in through the channel, or non-diffused, which means it just comes through automatically. There's no energy that's needed. Larger molecules will always need some sort of energy to be able to pass back and forth. It's called nuclear localization signal because it's dealing with the nucleus. Okay, it's not that it's radioactive or anything of that nature. It's just because you're dealing with the nucleus. All right, so the large molecules require nuclear localization signals to be able to pass through and get into the nucleus itself. So the nucleolus is the region inside the nucleus where the ribosomes are assembled from RNA and proteins. Okay, so here's where you see where the ribosomes are going to be put together. It's in the nucleolus. It's why it's kind of like the command center for basically life, because without these proteins, we would not be able to have and sustain life. The ribosomes, we already know what the ribosomes do, but they're made up of two different sized subunits. Okay, and again, they are slightly larger in the eukaryotes than they are in the prokaryotes. All right, and they're made up of that special RNA, that are ribosomal RNA and protein. That's what the ribosomes are made up of. And when protein synthesis is taking place, you can see here's messenger RNA coming in, it's reading it, transfer RNA is bringing the amino acids, and then ultimately the growing peptide chain is made. And you can see here's the smaller subunit, here's the larger subunit. It's the in between the two units is where the mRNA is being read. Okay, and then the transfer RNA is bringing in each amino acid to ultimately become a protein. It's during protein synthesis. Obviously, you already know this, the ribosomes assemble the amino acids into proteins, and it's in the third stage, or the tertiary stage, where the proteins actually become viable for use. The mitochondria, what is it? Powerhouse of the cell. It's what everybody's been taught since like second grade. The mitochondria is the powerhouse for the cell. It's where the conversion of stored energy takes place. Okay, and it's where ATP can be and will be created. This inner membrane is called the cristae. Okay, it's folded. Those folds are called the cristae, and the area enclosed within it, in here, the area enclosed in here, this blue, is called the mitochondrial matrix. Okay, so you have the mitochondria, kind of looks like, God bless, kind of looks like a long lima bean. You have the outer membrane of it right here. Here you have the outer membrane here. The outer membrane has to do with the mitochondria. The inner membrane has to do more with the cristae. It's inside. So the outer membrane is the mitochondria. The inner membrane is the cristae inside, okay? And it's in the mitochondrial matrix. That's what the cristae actually kind of encloses around itself. All right, peroxisomes. So the peroxisomes are usually very, very circular or spherical in nature. They are organelles that are enclosed by a single membrane, unlike the nucleus, unlike the cell itself. And what do the peroxisomes, their function is that they break down fatty acids, okay? And they also break down amino acids here as well. They also can take out poisons in the system, also known as alcohol. Bad kids. All right, so the peroxisomes may detoxify the poisons, they break down the fatty acids, and they also break down amino acids. All right, so animal and plant cells, what are the differences? Both of them have microtubules, but the animal cells also have what are called centrioles And that complex with the MTOC is called the centrosome, okay? Animal cells each have a centrosome and lysosomes, but plant cells do not. But plant cells have a cell wall, chloroplast, and other types of plastids, and a central vacuole, which removes water so the cells don't burst. Animal cells do not have that accommodation, okay? So the plant cells, that cell wall, very rigid, keeps the cell and allows the cell to keep its shape. Usually when you look at plant cells, they're square in nature, whereas regular cells are more roundish, ovally. And it's the chloroplasts and other specialized plastids that help the plant cells get their energy. And the animal cells with the cytosome and the lysosomes, it allows them to clear out the cells of poisons and things of that nature that are coming in. So when you look at an animal cell, here it is, okay? Very simple, looks very much like what we saw before. You have the secretory vesicles, which sometimes the cells will use to either bring something in or take something out, and usually it's something of a larger size that needs to come in. So, and we'll talk about what those types of bringing in and taking out are. And then plant cell, you can see more square in nature, a central vacuole. This, to be honest, in a plant cell is probably one of the most important organelles in the plant cell itself. Because the central vacuole, what it has is that it maintains the pressure against the cell wall. The cell wall wants to put in a plant cell, wants to push in the entire time. But this central vacuole is able to push back and keep that pressure where it needs to be so the cell keeps its shape. It does this by removing or bringing in water to the cell itself. Okay, and if there's too much water, what it will do is it'll take water out and expel it from the plant cell so it does not burst. And then if it's dry and it starts to shrivel, it starts to close in on itself, then it'll try to bring water in if it's available. All right. The cytoskeleton, again, giving it shape. The microtubules, intermediate filaments, and the microfilaments. Chloroplast, photosynthesis. That's where photosynthesis takes place. The plastids, they hold the pigments for the different parts of the plant itself. But you can see nucleus, nucleolus, endoplasmic reticulum. You can see the smooth part, not studded. Here, attached to the nucleus, the rough is studded, and those are the ribosomes. You have the Golgi apparatus, the mitochondria, okay, the plasmodesmata, which these connect to other cells. That's what you're looking at here, kind of these pores that are in there. The plasmodesmata is what connects plant cells to each other. All right. I'll go for another minute and a half. The centrosome consists of two centrioles. And what they do is they lie at right angles to each other. They form a perpendicular situation. And each centriole is a cylinder that has 9 triplets of microtubules. So you can see right here with the centrioles, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3. There's 9 of them that go all the way around, okay? And they are put together and attached to this cylinder, okay? And they're held together by these proteins. That's what these are. These proteins are kind of the glue that keeps the centrioles attached to the centrosome, okay? Those green proteins are a big piece. It's what ultimately holds it together, okay? So the whole thing is a centriole. This is, is a centrosome. I'm sorry. The whole thing is a centrosome. These are the centrioles, and these are the triplets that make up the cylinder itself and the microtubules. So the microtubules make up the triplets. The triplets, 9 of them, go all the way around, make up the centriole, and then the whole complex is the centrosome itself. All right. Questions about that? Yes, sure.
Where do you find all this? Like,
In the cell itself.
okay, so like when we're talking about the animal or the animal cell, where in the animal do you find that? Anywhere.
Everywhere. Everywhere there's a cell.
Okay, so even if it's like a skin cell, it's still
Yep.
considered an animal?
Uh-huh. Yep. Yes. Anything that is, so anything animal-wise would be,
Would that be us?
yes. Absolutely.
So like what are white blood cells? Is that considered an animal cell?
Yes.
Okay.
Yep. And we'll get more into that as we go further. All right. Any other questions? Good question. Any other questions? All right. Time's yours.